Hello, everyone. Thank you for the opportunity to present. I'm going to talk about using genetic constraint to improve the interpretation of missense variants and gene discovery. A recent big game changer for human genomic research is that now we can get access to large-scale sequencing data for general human populations.
Okay, sorry to interrupt. Can you share your screen? We are now seeing your slides, but not in presenter mode. Okay, go to the top down. There's a little present button.
Here.
Yep, right there.
Does that work?
And then can you go back to the Juno and then hit share?
But now I'm in full screen mode.
So after you hit the share, can you see your presentation popping up in the window in the entire screen?
Sorry, can you repeat that?
Yeah, so after you hit the present in presentation,
Yeah.
you go to the Juno and hit share.
Okay. Oh, okay. Now, when I choose the presenter mode, it will be in full screen.
And then you navigate back to the Juno.
Then I will exit the presenter mode.
Oh, can you do? Are you a Mac or PC?
Yes, it's a Mac.
Can you do a 3-finger swipe?
Let's see. Does it work?
Can you try presenting or navigate to the next slide?
Does it work?
We still see the first slide. Are you on the first or the second slide?
I'm on the second slide.
Another way is, can you do the entire screen, but click the first option, just share your full screen?
You mean when I choose to share in the platform, I choose to share the full screen? Does it work right now?
And can you try navigating to the next slide? Yep, that works.
Okay. Right, I will start from the beginning. So a recent big game changer for human genomic research is that now we can get access to large-scale sequencing data for general human population. And this has provided us a very powerful resource to study the effects of genetic variants, especially variants with large effects, variants that could severely reduce the reproductive fitness. Since they are under negative selection, thus we will assume this type of variants will occur less often compared with functionally neutral variants. Thus, we can quantify this removal of variants in our population against background expectation. And this is defined as genetic constraint. And this concept has been applied to prioritize disease genes, predict variant specificity, and evaluates important regulatory elements. However, since we don't have the power yet to evaluate constraint at single nucleotide level, we have to define a set of variants and aggregate the statistical signal over the set. For all this popular method I just mentioned, they try to aggregate the signal over the linear space of the genome. So in our work, we try to take a different angle and want to measure the genetic constraint over the orthogonal in the orthogonal space. And what does it mean for protein coding gene is we want to measure the genetic constraint over homologous residues. And we think we can aggregate the signal together since they are likely to have similar functional relevance. Thus, using this intuition, we have developed a novel score which measure the genetic intolerance of missense variation over homologous residue. And we name our method as homologous missense constraint, HMC. And here is what we do. First, we use PFAN database to define homologous residues. So if amino acids are in the same position of a protein domain family alignments, they are homologous to each other. And we want to measure the genetic constraint for each domain position for all human proteins with PFAN domains. To calculate the constraint for domain position, we need two quantity. One is the number of observed variants. This is directly counted from a reference population. And in our work, we use NOMAP version 2, which has 125,000 exomes as our reference population samples. For the other quantity, we need to know the number of expected variants. And for that, we have reconstructed a neutral mutational model from the NOMAD flagship paper. So it can give us the prediction based on 3-mer sequence contest, CpG methylation level, and sequencing coverage. Having these two quantity, we compare the ratio. So if the ratio of observed to expected is smaller than 1 for a domain position, it means this position is under constraint. And we further calculated the 95% confidence interval for this ratio and take the upper bound as our HMC score. So if HMC is smaller than 1, that means this position is under significant constraint. So following this process, we can map 28 million out of 70 million possible missense, rare missense variants in human genes with PFAN domains. And we took a stringent approach and only keep the domain position with good statistical power. So this gives us about 15 million possible missense variants accessible in about 10,000 genes. And we find that 22% of them, they are under significant constraint, and 9% of them, they are under high constraint. If we use a lower threshold, 0.8 to define that. And this is a value we find clinically relevant in our downstream validation. So after constructing the score, we ask whether HMC scores is associated with ClinVar classification. And as you can see, we do find that ClinVar pathogenic variants are significantly enriched in positions under constraints compared with benign variants. And this association is even stronger for precision under stronger constraints. In other words, variants with lower HMC scores are more likely to be disease-causing. And we also want to test whether this method is useful to prioritize de novo mutations. So we extracted published de novo mutations in cases with neurodevelopmental disorders and unaffected controls. And we find out that the cases do carry more constrained missense variants compared with the controls. Apart from using de novo mutations in healthy individuals as controls, we can also use a baseline de novo mutational model as our controls. So we can validate HMC by only using cases. And we did that by using a larger cohort of developmental disorders with 31,000 patients. And we find that in 285 genes related with developmental disorders, HMC-constrained de novo missense variants has a burden in cases comparable with the protein truncating variant. We also find that HMC scores could be helpful to study inherited conditions as well. And this is identified by seeing the association of constraint variants in sarcomere genes with cases of cardiomyopathies. Compared with the existing method, we know that a limitation of our approach is it can only apply to 20% of the exome. However, for the 20%, this method works. Whenever it scores a variant as highly constrained, its prediction is highly precise, and this precision is comparable with the existing method, including the one that is trained with supervised learning summarizing multiple computational scores. We also compare HMC, the distribution of HMC scores with two existing constraint metrics. And we find that it's not collinear with the other two. So that means HMC can add new information when it's used with the existing method. So having seen it's highly precise to prioritize de novo mutation, and also it's orthogonal to the existing method, we want to ask whether it can improve the discovery of disease genes. So for that, we have upgraded a recent published method called de novo West to test genes associated with developmental disorders. In upgrading, we incorporated HMC to score deleterious missense variants, and after upgrading the method, we find 7 newly significant genes associated with developmental disorders. And we find that for these new genes, they have more constrained missense variants than the protein truncating variants. Thus, it suggests these new genes, they're more likely to cause disease through autofunction mechanisms. And for 3 genes out of the 7, they are currently not included in the DDG panel. So this really highlights the potential of using HMC to improve gene discovery. So in summary, we have developed a novel metric, constraint metric, that measure the depletion of missense variants at homologous residue in reference human population. It is significantly associated with disease-causing variants, and we expect it's very especially valuable to prioritize the novel variants, as we can see that its prediction has an effect size comparable with protein truncating variants. And compared with the existing method, its precision is comparable with them, and also it's complementary with them. And we also demonstrate that it can improve gene discovery. We also learned the limitation of our approach. Currently, it can also assess 20% of the exome, but this approach can be scaled up when we have more exome sequencing data in the future. We also know that this method, the accuracy is highly depends on how we define the homologous residues. As we are measuring negative selection, we wouldn't expect this method directly work well for late onset disease. For more details about this work, we have a preprint available at MacHive. And at the end, I would also like to thank my supervisors, my colleagues and collaborators, and also patients and other volunteers who have contributed to the research. And thank you for your attention.
All right. Thank you, Xiaolei. As our next speaker, Xiaolei loads her slides, or his slides, sorry. Does there maybe a very quick question? Okay, I think Antonio has one. Please go ahead.
Thanks for the nice talk. Have you checked how it relates to interspecies conservation, and eventually have you disentangled the performance to identify disease variants according whether the position is conserved across the species or just constrained within human?
We haven't did that validation because, as I have mentioned, the big limitation we know is it can only apply to 20% of the exome. So yeah, we haven't really looked at that.
All right, thanks.